Hello everybody. In chapter 8 we are going to talk about DNA, RNA and protein synthesis. Section 1 we talk about in section 1 we are talking about discovery of the DNA. From his study of pea plants, Mendel concluded that heredity factors determine many of an organism's traits. Previously, that it has been proven by Mendel that the factors control traits. Okay, but what were these heredity factor? Now, the question is that what is this heredity factor? How did this molecule store heredity information? For example, the color of the eye is a heredity information. How the factor store the heredity information that's related to the eyes? How did this molecule store heredity information? Scientists believe that if they could answer this question, they would understand how chromosomes function in heredity. The answer to this question begins to emerge during an epidemic of pneumonia in London in 1920s, because at that time in London in 1920, the disease pneumonia appeared, and the scientists worked on the pneumonia. They tried to find a vaccine for it. So during their study, they 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 found something about DNA, RNA, DNA, RNA, especially DNA. Now, first scientists who work on this, it was Griffith. So Frederick Griffith is a scientist who studied this bacteria. So Griffith in 1928, he was a British medical officer. He was studying a bacterium named Streptococcus pneumoniae, abbreviated as S. pneumoniae. Some type of or strain of this bacteria can cause lung disease, pneumonia in mammals. Griffith was trying to develop a vaccine against disease caused or virulent strain of the bacteria. So the virulent strain, so we have two strains. One of them is virulent and the other one does not cause disease or avirulent. So he tried to find a vaccine for the virulent one, two bacteria with the same species, but two strains. The virulent bacterium is surrounded by what? A capsule. And mostly the bacteria who is covered with the capsule, they are protected from the drugs from the immune system of the human. So surrounded by what? A capsule. This capsule is made of polysaccharides that protect it from the body's defense system. The bacteria in a virulent strain grow as a smooth-edged colonies, then grow on petri dish and called the S strain. In contrast, the second strain of Streptococcus pneumoniae does not cause pneumonia and lacks a capsule. The second strain is called R strain because it grows into rough colonies. The R stands for the rough. So the R strain also shown in figure 8.1. Look at the figure 8.1. Let's move to the figure 8.1. This is the figure. So you see in figure 8.1, we have Griffith study Streptococcus pneumoniae bacteria. The S strain can cause pneumonia. The R strain does not cause them. So see, one of them has smooth edged, which is as S strain, which causes the disease. The R strain does not cause disease. They have rough edges when they are grown in petri dish. Now, this is the difference between Streptococcus pneumoniae as S strain and R strain. And the S strain is harmful. The R strain is harmless. S strain is surrounded by capsule, but R strain lacks capsule. Cause lung no, in the S strain grow in as a smooth-edged colonies, while the R strain it grows in rough colonies, cause the lung diseases, pneumonia in mammals. This is S strain, but R strain does not cause the disease. So this is the comparison between S strain and R strain. Now let's move to the experiment, the Griffith's experiment. This is the Griffith experiment. What he did, he do four experiments, or he did four experiments. In experiment one, he injected mouse with the R cell. R cell, we said that is a rough which have rough colony, the bacteria, Streptococcus pneumoniae bacteria, which have rough colony. This one is harmless. So because they are harmless, the result does not cause anything to the mouse. So arsenic do not kill the mouse. But in the second experiment, we see that Griffith inject mouse with the live cell, and we know that the cell is kills the mouse, so the mouse will die. In experiment 3, he killed S cells with the heat, injected mouse with the heat, killed S cells. Then the killed S cell injected to the mouse, so the mouse does not die, so does not kill the mouse, because the S cell has been heated. When you heat bacteria, the bacteria is going to be ruptured in different parts. In experiment 4, he killed S cell with the heat, as experiment 3, but he mixed it with the live R cell. We know that the heat S cell does not kill the mouse alone, and also live R cell alone does not kill the mouse. But when he mixed them, he injected mouse with these two, he killed the mouse. So the mouse will die. So something happened. Why this is happening? So in his fourth experiment, he injected mice with both heat killed S cells and live R cells. He found that the mice died. Why the mouse died? Griffith concluded from his fourth experiment that heat killed parent bacteria cells release a hereditary factor that transfers the disease-causing ability to the live harmless cell. Okay, so this is what we can do there. Let's see. Type of transformation. So some transformation. The type of this transformation. This type of transfer of genetic material from one cell to another cell, or from one organism to another organism, is called transfusion. Okay, so this is what's happening. That was transformations. Transformation happened, not transfusion. So transformation. What is transformed? How this transformation happened? This is what gives the conclusion. To answer what is transformed or how the transformation happened, another scientist named Avery came found that how this transformation and the substance that transformed uh, uh, what's the substance and what's the substance that has been transformed. So he do an experiment again. What he did, he repeated the fourth experiment 3 times. He added an enzyme to destroy different molecules. For example, in the experiment 1, so he repeated the third experiment of the Greek experiment, fourth experiment of the Greek experiment, and repeated them 3 times. In experiment 1, he bring heat killed S cells and he added protease to it. It means that the heat killed S cells, it is die and the protein digested, so it does not contain protein. Then he mixed with the live R cells. He injected mouse with the mixture. He found that the mouse was killed. Then in experiment 2, he add RNAs. So in this time, the heat killed S cells does not contain RNA. It means there's protein and DNA is there. And we add RNAs because RNA is an enzyme that destroys RNA. DNA is an enzyme that destroys DNA. Protein is an enzyme that destroys protein. Okay. Then in the experiment 2, heat killed S cells plus RNAs. So this heat killed S cell does not contain RNA. Then mix it with what? Live air cells. Injected with the mixture. The mouse will die. So till now, no protein, neither protein nor RNA are healthy materials. In experiment 3, he brought heat killed S cells and he added DNAs. So this heat killed S cells was not contain DNA, mixed with the live R cells. Then injected mouse with the mixture. Does not kill the mouse. It means because lacking DNA, the transformation does not happen. If lacking of the RNA transformation will happen, lacking protein transformation will happen. But lacking RNA transformation does not happen. This means that RNA or DNA is a hereditary material. So what the other conclusion is that the DNA is a hereditary material in bacteria. Now we have another experiment, Hershey and Chase experiment. These two scientists, Martha Hershey and Martha Chase and Alfred Hershey, set out an experiment to test whether DNA or protein was the hereditary materials. Viruses transfer when virus enters bacteria. Virus that infect bacteria are called bacteriophages. No, there's a virus. Virus does not only attack human, also attacking what? Attacking bacteria. The one who's attacking bacteria called bacteriophage, or in just a phage. So what he did, he labeled the virus. He did both of them do they do an experiment. In experiment 1 and experiment 2, they labeled the virus. One of them, the code or the protein code of the virus, has been labeled with the radioactive isotope S35, and the other one, the DNA, is labeled with the radioactive isotope P32. So they are radiated. You can detect them under the machines. So what he did in the first experiment, label bacteriophage with the radioactive isotopes, allow phage to infect bacteria cells. In the second step, he used blender to remove viruses code from the surface of bacterial cells. So the virus code is removed. He centrifuged to separate heavier bacteria from the lighter phage. Note that the bacteria is heavier than the phage. S35 radioactive did not enter bacterial cells, so it does not go to bacteria. So S32, whose label is the protein label to the S35. So the S35 does not go to the bacteria. It means the protein does not go to the bacteria. But whose go with the bacteria is P32, radioactive enters bacterial cells. It means, so whose label with the P? It is DNA. So DNA goes with the bacteria. So who's the hereditary materials? DNA is a hereditary materials. This is the conclusion of these two scientists. Thank you.